and it is noon East Coast time. So hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani, and I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. With me today from NCCAT is Ed Eng, and joining from the Pacific Northwest Cryo-EM Center is Vamsi Rayaprulu. And from the Stanford SLAC Cryo-EM Center S2C2 are Nathan Burrows and Michael Schmidt. PNCC has invited today's speaker, Kirill Nadezhdin, who's joining us from Columbia University. And Vamsi will introduce him properly after a quick update from each of the 3 centers to keep you up to date on the resources we make available at no cost to the research community. This webinar is a regular event that we host to highlight the methods and tips and tricks that groups are using to collect and interpret data they can obtain with the help of National Center resources. Excuse me just a second. We usually meet the last Thursday of the month, but to make sure that we don't get in the way of anyone's turkey or other holiday celebrations, we will not have a talk in November, and in December we'll meet a slightly earlier date, so you can save the date for December 14th. And instead of a regular talk, we'll be hosting a roundtable with a panel of staff microscopists from all of the centers. This will be targeted especially to new practitioners who, if you've ever stared at gray images coming from a microscope and wondered, what is it I'm looking at? Or is Cryo-EM just a cruel game of magic I don't know how to play? We encourage you to come join us in December, and we're going to look at a collection of interesting and probably also not so interesting micrographs and answer the question, do you see what we see? So this will be a more interactive session. We'll be setting up a way for you to pre-submit images that may be baffling you. And so we hope that you can come join us in that conversation. The registration link will be available on the events page at cryoemcenters.org soon, so look out for that. We hope that will be a fun time in December. So back to today. For Zoom logistics, please send your questions at any time during the talk using the Q&A box. We'll be watching there and not in the chat. And Vamsi will be moderating questions at the end of the talk. And with that, I will turn over to our center representatives to let you know what's happening and what's new at the centers. So Nathan, why don't you go ahead first?
Hi, so I'm Nathan Burrows. I'm the screening program manager at S2C2, and we have lots of exciting things going on at our center. The latest is that we just got our VitroJet installed and commissioned, and it's working. So we now have a VitroJet for preparing samples in addition to a Chameleon and the standard VitroBots and Leica EM GP2. We also are excited that we currently have 3 microscopes on site that are in queue to be installed and commissioned. We're adding another Krios, a Glacios, and a Tundra. And so we hope to have all those set up and operational probably by the end of this quarter, if not next quarter. So we're all very excited about that. So lots of things moving around here at S2C2.
All right, thank you, Nathan. And I'll turn over to Ed.
Hi, I'm Ed here in New York. We offer a high-end instrumentation access program on our 4 operational Krios instruments. We offer a robust screening program where you can make use of our plunge freezers, a Chameleon, our Tundra, and Glacios. On that front, we've just received our 2nd Glacios, and we're installing that over the course of this winter. So hopefully we can bulk up that screening capacity. And training, we offer a very robust cross-training program. A couple of things happening next month. We'll have a micro-AD workshop. And this spring, we're going to have our single particle short course. So there's still room for a single particle short course. Please apply. For more information, go to nccat.nyspc.org.
Thank you, Ed. And Vamsi, go ahead.
Hey, everyone. I'm Vamsi Rayaprulu, and I'm a microscopist at PNCC in Portland, Oregon. A quick intro into what we offer here at PNCC for researchers. We have two types of proposals. The first one is a screening access proposal, which allows you to come on site for up to 5 days of sample preparation and screening with staff. So you'll be on site just doing all the things and learning all the necessary information about grid freezing and screening and data collection. This proposal is aimed at low resource institutions. The second type is called a general access proposal, which awards up to 480 hours of microscope time a year for 2 years, so a total of 960 hours. This time can be used for screening or data collection. Approved general access proposals are also eligible for our training workshops, which cover topics ranging from grid preparation to all the way to data processing. In both cases, approved proposals are assigned a scientific point of contact, a SPOC, basically me, or a microscopist like me, who can help you with questions about sample optimization, data collection strategies, and so on. To help with our data collections and screenings, PNCC has an Arctica and 4 Cryoses with all with K3 cameras. 3 of our 4 Cryoses have energy filters, while 2 of them also have a Falcon 4i. For sample preparation, we have a couple of Vitrobots, a Leica GP2, and for the Brave, a manual plunger also. If you're interested in applying, our deadline is the 1st of every month. We look forward to hearing from more of you. We've been getting a lot of proposals recently, and we love that. With that, let me introduce our speaker for today, Dr. Kirill Nadezhdin. Kirill completed his undergraduate studies at Moscow Institute of Physics and Technology, where he focused on mathematics and physics with applications to biological systems. He earned his PhD in biophysics from Lomonosov Moscow State University while researching the amyloid precursor protein associated with Alzheimer's. His specialization was in NMR methods applied to solving high-resolution structures of short membrane proteins. After completing his PhD, Kirill continued his work in the field of structural biology using NMR to solve the structures of transmembrane domains of neurotrophin receptors and biologically active peptides. He worked as a research scientist at the Institute of Bioorganic Chemistry in Moscow, Russia, in the laboratory led by Professor Alexander Arseniev. In 2019, Kirill transitioned from NMR to Cryo-EM. Thank you, Kirill, and joined Dr. Alexander Sobolowski's lab at Columbia University. He now specializes in studying the structures of ion channels using Cryo-EM with a particular focus on transient receptor potential TRP channels, specifically the temperature-sensitive TRP channels, and has recently published several exciting articles. Today, he's going to talk about his work on TRPM7 channel, the master regulator of divalent cations in the human body. Over to you, Kirill.
Thank you very much for this kind introduction. Please let me try to share my screen. Okay. I think you can see my screen right now, right?
Yeah, looks good.
I want to hide all other distractions. Yeah, should work now. Okay, so let's start. So hello everyone, I'm Kirill Nadezhdin, and for the last 4 years I've been affiliated with the Sobolowski lab at Columbia University in New York, where I've been investigating the fascinating topic of structural mechanism of TRP channels function, activation, and inhibition. And today I'm excited to share some of our latest findings in this area. And today I'm going to focus on TRPM7 channel, which is unfortunately not temperature sensitive, but it is a very cool target to work on. So let's dive in. So, but first I want to start with acknowledgements, because sometimes there was not enough time to thank all the people who helped me with this work. So this work was split into 3 labs. Most and all of the structural studies was done at Sobolowski's lab at Columbia University in New York. But a lot of things were done for this particular project in Munich in the lab of Vladimir Chubanov, who did 90+% of all physiological recordings for this paper were like crucial for us. And also we did some MD simulations for the study to confirm the results that we got from the Cryo-EM. And that was done in Carnegie Mellon University in Pittsburgh in the lab of Maria Kornikova by Chamali and Dillon. I also want to thank agencies that helped to fund this research, and Cryo-EM facility at Columbia University, Jening and Surajit, who worked at that time when we did the studies, and Bob. And also, I want to thank a lot all the facilities where we collected our data. And we had really good experience with working with all the facility, SLAC, the NCC, NYSBC, and we are very thankful for all the facility who help us to collect this beautiful data. So in our lab, our group that I work with, we are mostly focused on transient receptor potential channels, so TRP channels or TRP channels. And those are very interesting objects to study. Why? Because there are 28 TRP channels in mammals, and 11 of them are temperature sensitive. And they are both heat or warmth temperature sensitive, or cold or noxious cold or cool channels. I want to show my laser pointer. Yeah. So, and those are not just temperature sensitive channels. They are actually the sensors of heat and cold in human bodies. And they're also interesting targets to work on because they not just sense temperature, but they also can be activated by a lot of small molecules and compounds that can be found in food. So, for example, if you like spicy food, you can feel this burning sensation in your mouth when you eat something that contains capsaicin that can be found in a lot of chili peppers and a lot of like these hot peppers. Or you can also feel this cooling sensation when you, let's say, drink some refreshing drink that has menthol in it, or brush your teeth, or chew bubble gum. That can, and this menthol, it activates one of the TRP channels called TRPM8. But today, I'm gonna talk about not temperature sensitive channel. I'm gonna talk about one of the member of melastatin family of the TRP channels. So this picture was taken from the just published review from our colleague from Munich, Vladimir Chubanov. This is a very beautiful work. So thank you to him that he provided us with these beautiful illustrations. So the TRPM family, melastatin family of TRP channels is involved in a lot of inherited human disease and consists of 8 members. And 4 of them are temperature sensitive and 4 of them not. I'm going to talk today about TRPM7 channel that is considered to be the master regulator of divalent cations in human bodies, and it conducts a lot of divalent cations. And what is interesting about this particular channel and its highly homologous TRPM6 channels, they have this kinase domain that has kinetic activity and do a lot of stuff in human body apart from just being a channel. And TRPM7 channel is involved in rare inherited disease like macrothrombocytopenia. This is a rare platelet disorder characterized by a reduction of platelet counts and abnormally large platelets. And TRPM6 and TRPM7 channel, they are also involved in primary hypomagnesemia. This is an electrolyte disbalance caused by low magnesium levels in blood serum. So although maybe it's not so severe, may not be so severe, but let's talk a little bit about the minerals in human body. So our body relies on minerals, and it cannot produce any of these metals. So we have to eat food and drink a liquid to obtain all these minerals. And the most abundant one is calcium, then is followed by potassium, sodium, magnesium, a lot of iron, zinc, copper. And it is interesting that the intracellular levels of these minerals is not the same. And you can see, like, for example, magnesium, there is a lot of magnesium inside the human cell. And it is the most abundant divalent cation, magnesium, the most abundant divalent cation inside the cell. So all these minerals, they do a lot of functions, and they are involved in a lot of reactions inside human body, but they actually are different by their physical nature. So all these metal ions have different radii or different charge. So that translates to the charge density. And I'm not going to talk about the iron today because iron does a lot of stuff in human body, but mostly it is involved in oxygen delivery. But if you look a little bit closer, the charge density of magnesium, zinc, and copper is quite high compared to more abundant sodium, potassium, and calcium. It means, so this translates to that those minerals are really involved in a lot of biochemical reactions in human body because of this high charge density. It is beneficial for the cells to use these ions for chemical reactions. And just, for example, for magnesium. So magnesium forms complexes with proteins, DNA, RNA, and for example, if you want to run PCR, it will not work if you don't supply magnesium. Or all the ribosomes, they have a lot of magnesium bound to the complex. And again, magnesium participates in hundreds of biochemical reactions in cell, including energy production, protein synthesis, and yada, yada, yada. So talking about tryptophan-7 channel. So tryptophan-7 is the gatekeeper of mineral absorption of these minerals from food in our bodies. And it is considered to be one of the most, even not the most important channel for the mineral absorption of zinc, magnesium, and calcium inside the cell. And the heterocomplex of highly homologous tryptophan-6 protein with tryptophan-7 also conducts a lot of magnesium. Interestingly, that tryptophan-7 channel is also inhibited by magnesium and magnesium ATP inside the cell. So there is a kind of a loop that prevents the overload of the cell by magnesium and other divalent cations. So for this project, we use the kinase domain truncated version of the channel that was shown to be fully functional in physiological recordings. And this is just brief overview of how we express our proteins. So we produce plasmids, then we transform E. coli to produce backmids. After that, we use backmids to transfect SF9 insect cells. And after transfection, these cells produce the virus, baculovirus. And after that, we use this virus to infect HEK mammalian cells to produce protein. And this is just a little bit like more detailed version of it. So if we start with SF9 insect cell transfection, so we use backmid and transfect SF9 cells to produce the low titer of virus, very low concentration. Then we harvest virus and we use this virus to infect again SF9 cells to produce even more virus in large scale. After that, we concentrate this virus and use the concentrated virus to infect HEK mammalian cells, express the protein for a couple of days, and pellet them and store them for protein expression. And talking about the protein purification, and talking about the protein purification, this is the overview of what we do usually when we purify our protein. So we lyse the cells, usually using ultrasound. And after that, we centrifuge to collate the membrane fraction of the cells. After then, we homogenize these membranes and add detergent to extract protein from the membrane fraction. When the protein, after the protein is extracted by the detergent, and in this case, we use GDN, but I will talk about this a little bit later. We add resin and have, and after that, it is followed by two-step purification. First by affinity tag chromatography, and after that, site exclusion chromatography to purify from aggregates, other proteins. And after that, we have pure protein that we apply to grids and do Cryo-EM. So fine. So yeah, we'll have this slide. So after protein purification, we do Cryo-EM. So let's just see like the real data. So this is the profiles from site exclusion chromatography, and we, for protein purification, we use, for protein extraction from the membrane fraction, we use the detergent GDN. And sometimes if we need to, we like to reconstitute our protein into nanodiscs. And for this particular project, we use MSP 2 nanodiscs. And this is the typical micrograph that we got for this kind of for our grids. It is really good. We have no issues with the distribution of particles throughout the micrographs, and the 2D classes look just fantastic. So, and we have no issues with picking the particles and alignment of the particles. And now comes the data processing. So for this particular project and for many projects, we use some kind of different approach that usually people do for processing of their data. I prefer to use both Reliant and CryoSpark at the set for every data set, because in my experience, I get a little bit better resolution each time. So everything starts with the motion correction in Reliant. And why I do this, because later on I want to do Bayesian polishing in Reliant. So it's easier for me to do motion correction in Reliant. And after that, I transfer all the micrographs into CryoSpark. Why I do this? Because again, in my experience, it is way more efficient to continue processing of motion corrected micrographs in CryoSpark. I do the CTF estimation, blob picking, 2D classification to create templates, and template picking works perfectly fine for this protein, and I extract particles. After that, I do multiple rounds of 2D and 3D classification to remove junk particles again in CryoSpark. And why I do this? Because so we do multiple rounds of 2D and 3D classification in CryoSpark. And why we do this? Because it works way faster for us in CryoSpark than in Reliant, and we also rely mostly on 3D classification to remove junk particles, because we end up with more views into the classification. And it looks like when we do just 2D classification, sometimes we can just get rid of some rare oblique views. So after this small round of 2D, 3D classification, we end up in 500,000 particles, almost 600 particles, and the resolution is about 4 angstrom when we run refinement, when we import everything back into Reliant after this junk removal. And the next step is done in Reliant. We do 3D classification. I do 3D classification in Reliant. After that, I pick the classes that look the best looking classes that are have most of the features. And after that, I re-extract particles without binning in this case, and I do CTF refinement, Bayesian polishing, and second round CTF refinement. And by this time, we already reaching the resolution of 3 angstrom without masking compared to about 4 angstrom before 3D classification and all the CTF refinement Bayesian polishing steps. So next, one more round of 3D classification in Reliant, again, picking the best classes, do Bayesian polishing the second time in Reliant. After that, I import particles back to CryoSpark, run refinements and non-uniform refinements now with C4 asymmetry imposed, and we get like really beautiful structure of close to 2 angstrom, 2.19 angstrom resolution that looks like this. And the quality of the map is excellent, and we see a lot of features. We can easily build the model, but if you look closer to this map, you can see that although the upper part of this channel is resolved like really well, and the resolution is about, here is about like 2.5, you can see all the lipids and all the side chains. The lower part of the channel, the intracellular part, it isn't resolved like really well. You can see that the resolution is here, the estimated resolution is close to like 3.5, even like 4, even worse. It depends on which threshold you like. So why it's happening? Actually, so when we run variability analysis in CryoSpark, we can see a lot of movements in the lower part of the channel. So if you kind of disregard this micelle flickering back and forth, you can see that the transmembrane domain of the channel stays intact. So it is not affected at all, but the lower part of the channel actually moves a lot. And you can see this like flickering between like 2 diamond shapes of the lower part. This is the bottom view of the channel. And this is the one mode. And this is an example of another mode from variability analysis from CryoSpark. And here you can see some different movements. Again, you can see these movements in the lower part. Nothing actually is happening in the transmembrane domain. And you also kind of see that there is a cold coil domain appearing and disappearing if you go back and forth. So because of this variability, because of these movements and heterogeneity in our sample, we can see that the lower part of the channel isn't resolved really well. So what we can do, of course, we can go back to our processing, and what I did, I created a mask around the N-terminal region. This is N-terminal region of the channel, the lower part of the channel. And after that, I symmetry expanded our particles, did particle subtraction, did 3D classification with a mask around this region, local refinement, and after that I got this very well resolved part of the 2.7 channel, the lower part of 2.7 channel, or at the resolution better than 3 angstrom. And after that we combine both the upper map for the whole channel and the lower part for these MHR regions in one final map in Phoenix. And the resolution of the global uniform refinement is close to 2, and the resolution of local refinement of the lower part is close to 3. So what we got from this local refinement, actually everything that you can see here on the left in green, this is the new build stuff that we were able to squeeze and build the model because of this local refinement and symmetry expansion and local refinement. But interestingly, this is just one subunit of chip channel that consists of 4 subunits. But interestingly, so that was not the first structure that was published on 2.7 channel. Back in 2018, the first structure and the only paper about the structure of 2.7 channel came from the David Clapham lab. And they solved the 2.7 channel, same truncated construct. They didn't see these extra parts, basically, most probably because the software was not well developed as it is right now. And interestingly, one more interesting thing is that although they tried different conditions, adding different minerals, different compounds to the, they tried different conditions to make the samples, they end up with 3 structures. They look absolutely the same to me, and they also look absolutely the same to the conformation that we got. So this is kind of the observation how it looks like. So this is the map, the full map. You can see the kind of micelle or nanodisk wraps around. The quality of the reconstruction is very good. We see a lot of lipids that wrap the transmembrane domain that is here. And interestingly, we also can spot some, so we can assign some densities that we think are lipids to phosphatidylcholines, cholesterol, and also GDN. This is the detergent that we use to extract our protein from the cell membrane. And I want to talk a little, just one slide about the cholesterol, because I found it like really fascinating, because it it goes back to my memories from my university, from biochemical class. So this is, we see cholesterol, and it resides somewhere here in transmembrane domain. Interestingly, for cholesterol, that we never added and we never had cholesterol present in our buffers when we extracted the protein, when we purified it. We also reconstituted this particular sample into nanodiscs. When we did this nanodisk reconstitution, we didn't add any cholesterol to our lipid mix, but still we can see cholesterol bound to the channel. So it means that this cholesterol is endogenously survived. So it is endogenous cholesterol that came from the cell, and it survived bound to tryptophan-7 channel during all steps of protein purification and reconstitution into nanodiscs. And one also interesting thing, the quality of the reconstruction is so good. So actually, so this should be the video, but it doesn't play. So let's then just skip it. So interestingly, so when you can see the shape and the map of the cholesterol, it actually resembles what I was taught in my university. So one side of the cholesterol is completely flat, and you can see, although it's not playing. So can I play this video somehow? I think I cannot. So it doesn't really matter. Then, so you can see that the other part of the cholesterol has two bumps from methyl groups. So it is like very cool to observe. So it kind of justifies that the professor that taught you about the biochem class, he was right. So we end up solving the structure of tryptophan-7 channel in close conformation that is absolutely the same to the structure that was solved before in 2018. So it was not very interesting. So we decided to do something to move the conformation of the channel to some open state. And luckily we have published gain of function mutation when asparagine is substituted by glutamine. And as you can see, if you introduce this mutation, the current through the channel is way higher compared to the wild type, about like 10 times more in patch clamp recordings. But does it happen for our construct? Actually, it does. So this is single channel recordings from the purified channel that we used in Cryo-EM and reconstituted into planar artificial membranes. So this is single channel. So just one channel is inserted, and you can see the current through the just like one particular channel. And for the tryptophan-7 channel, without any ligands, for the APO wild type state, without any mutations, you can see some kind of sporadic flickering of the channel, and it was expected because this channel is so-called constitutively open. So it means it kind of opens and closes spontaneously. But when we introduce this mutation, we can see that the opening happening is like way more. You can see it, and you also have these long stretches of the open channel, for example, here, where it stays open for almost like 1 minute or even like more than 1 minute. So we solve the structure of this channel with this mutation, and interestingly, that as expected, The channel now isn't in open state, although it looks like very similar. So we see some differences only in the pore of the means. So everything stays the same mostly, but only pore becomes wider. And you can see here also on the pore profile. One thing that I want to mention here, because it's going to be important later, that all particles were in this particular state that we consider to be open. So there was no like heterogeneity between closed and open state, like all particles in Cryo-EM conditions were in open state. So this is like more obvious when you compare the closed state, the pore, indeed, it is wider. It is wider, but is it open? So for that, we actually asked our colleagues, we approached our colleagues to run MD simulations for us. And this was done in Carnegie Mellon University in the group of Maria Kurnikova. So they used transmembrane domain, our structure, and they ran MD simulations to see if there is any conductance through the pore. And for the apo state without any ligands, without any mutations, you can see that water cannot pass through this constriction caused by isoleucines. And also, ions cannot go through these through the pore. But for the mutated state with the mutation, you can see that water can go through the channel and also ions can go. So this was kind of independent justification that indeed our pore is open and it can conduct ions. So now we're kind of, it was confirmed that what we got in Cryo-EM was indeed the open conformation. So this is again the video, but I don't think it's going to work right now. So what they basically did, they show, so there's a video that shows how ions can pass through the pore under the applied voltage. Okay, so this is good, but this mutation is not physiological. So it doesn't happen in like any like natural environment. So this is artificial mutant that can open the channel. So who cares? So that's why we decided to see, okay, can we open the channel by something else? And for example, the ligand activator or agonist of TRPM7 channel. Luckily, we have a list of them, and this is one of the most used agonists for TRPM7 to study TRPM7. And again, the colleagues from Munich, they showed in page clamp recordings that indeed, when you add naltriben agonist, there's like way higher current through the channel. And again, I also did these single channel recordings for the protein without adding naltriben and after adding naltriben. And as you can see, there is like a huge difference between these two recordings. So when I added naltriben, you can see a lot of open state conformation, and they last for like several seconds. Okay, so we saw the structure of 3.7 channel in complex with this agonist naltriben, and we can see it where it binds. One thing that is actually very important here, we tried to solve the structure of the open state in detergent. I think it didn't work for us. So for this particular open state, we had to use nanodiscs. And it gave us like pretty good resolution. And as you know, for nanodiscs, you need to have like enough protein to reconstitute your protein after purification into nanodiscs. So this is kind of the trade-off. And kind of I want to mention that apo state in nanodisc and detergent looks completely the same. But again, in nanodisc, when we added naltriben, it didn't open the channel. So we see that naltriben binds. We see where it binds. The map quality for this ligand is very good. So there's no doubt this is indeed this molecule. And what we see, what naltriben does, it's actually glues two subunits together. So this is like one subunit, 104, that is colored in gray. And this is another subunit colored in pink. And actually, this naltriben works as the glue. It kind of pulls two subunits closer to each other, because without naltriben, they really far apart, like several angstroms apart from each other. So again, the pore is much wider than in the closed state, and our colleagues who run in these simulations, they also confirm this is indeed the open state. It can conduct ions and water. And we also, with the help of our colleagues, they did a lot of functional studies that I cannot show you because this topic is mostly focused on cryo-EM and structural biology, but they did a lot of work on this work, a lot of functional work. So they did all the mutagenesis around this site. And basically, if you touch any residue that is close to the naltriben binding site, you can see the reduction of TRP activation. And if you do even like double mutation, you can see even more reduction in TRP activation by naltriben. So these functional studies, these mutagenesis confirm that, yes, this is indeed the naltriben binding site. Unfortunately, I have a video that I cannot play here, so I will skip it. One very interesting thing happens to this data set. So I didn't mention during processing for the data set when we added naltriben, we actually got another state for the naltriben binding. That was completely different from the open state when the ligand was bound. Interestingly, that was like very surprising for us, is that the naltriben binds in completely different spot. So basically, it looks like that when naltriben finds TRPM7 channel, it has two choices: either go to one side or go to another side. We didn't see any overlap of binding. Let's say there's a tetramer, so we should expect like 4 ligands bound to the channel. So we didn't see, let's say, like 2 bound to one side and 2 bound to another side. No, it's kind of black or white. Either 4 spots are occupied and channel is open, activated by binding site, or other 2 spots are occupied and the channel is closed. And that was kind of interesting. So one of the reviewers asked us to explain why it's happening. We actually don't know, but at least when we try to compare the densities for naltriben binding in both spots, in this spot and in this spot, the densities look the same, and the model for that it was fit into this density was also the same. So it looks like that that naltriben doesn't change its conformation. It just kind of chooses between 2 binding sites. And this binding site resembled the apostate very much. I mean, the binding site where naltriben bound and the channel is not open. So next, we decided to see, so can we inhibit the channel? So can we find the site for TRPM7 inhibition? Because we know the binding site for activation, we have some weird site where basically it does nothing, but can we find the site where we can inhibit the channel? And luckily, again, we have a lot of compounds that can be tested. We choose the most used one in the field of TRPM7 physiology. We call these compounds VER and NS. And as you can see here, when you add these compounds, the current through the channel in patch clamp recordings is way lower. So yeah, patch clamp recordings confirm that yes, these are indeed inhibitors. And we added these inhibitors to our samples in Cryo-EM, and we found that although the nature and the chemical structure of these 2 compounds are different, they all bind in the same spot here. And this is different from the activating binding site for naltriben. And the pore is closed, and the state resembles, again, this state with inhibitors bound was very similar to the apostate without any ligand. So we need to have some confirmation. Okay, so maybe So this is like indeed ligand binding, specific ligand binding, but not just kind of this small molecule finds a groove, hydrophobic groove somewhere in the transmembrane domain of the channel, it just resides there. So again, our colleagues from Munich, they did mutagenesis, and they indeed confirmed that when they mutated the residues around the binding site of both these ligands, indeed you can see the shift of the tryptophan activation curve. It means that yes, they do bind here. And as you can see, they really look different. So they kind of no homology between them, like at all. And the quality of the reconstruction is very good. So, now comes the interesting thing, one more interesting twist, that the binding site for this ligand, naltriben, that was not the activating site, basically overlaps with the binding site of these two inhibitors. So it means that we found the kind of universal binding site site for inhibition of tryptophan channel. And this is kind of the full list of structures that we have from this project, and I want you to pay your attention to these two structures. Why? Because again, there was kind of the problem to justify that indeed when we add ligand, it actually binds to its specific site. So, and as our apostate and ligand bound state are the same, we have to have more proof that indeed the ligand binds to its specific site. So for that, we also used the mutation, the mutant for tryptophan. So as you may remember, I told you before that when we saw the structure of the mutant, we see all the particles were in this open state. But when we add ligand, all the particles now in the inhibited state. And this is true for both this compound and this compound. So we have kind of different justifications that indeed, yes, this is the ligand binding site, this is the inhibitory binding site. So in this, after that, we end up with this model of tryptophan activation and inhibition. Tryptophan channel usually sits in its resting closed state, but this is constitutively open channel. So sometimes it opens, and we speculate that when it's spontaneously open, it resembles the conformation that we got for the mutation. Why we think so? Because the movements happen only in the small portion of the ion channel pore. But if the cell needs magnesium or zinc, it recruits some endogenous compounds to activate the channel. And we can see a lot of movements in the lower part of the channel. Unfortunately, my video didn't work, so you didn't see this, like a lot of movements in the lower part of the channel. So the agonist bound state differs a lot from the spontaneously open channel. And when there are too much magnesium or too much divalent cations inside the cell may decide to inhibit the channel by the means of, let's say, magnesium or other like magnesium ATP that can bound into this inhibition site. So the activation site and inhibitor binding site, they're different. And there are two different open states for tryptophan channel. So this is kind of the full diagram of the tryptophan activation and inhibition. So, and with this, I apologize that my videos didn't work, so I didn't have a chance to show you. And with this, I want to thank you for your attention. And I just want to keep this slide. The Sobolevsky Lab is hiring. So if you want to join the Sobolevsky Lab, please write an email to Dr. Sobolevsky to discuss the opportunity to join. Thank you.
Thank you, Kirill.
Yeah, thank you so much. That was absolutely amazing. Since we are running up on the hour, I want to say for those of you who have to run off, thank you for joining us. We are going to stick around maybe a few extra minutes to make sure we get to all the questions that have already been sent in and are coming in. So if you do have to run off, we will post the recording, including getting to the rest of the questions. But if you do have to run away, thanks for joining us, and we hope to see you in December. So Vamsi, I'm going to let you start moderating Q&A.
Sure. There are a few questions in the Q&A section. The first one is, hi, is the virus production from a back-to-back system like a PFAS back?
So we use different system. It's not PFAS back. We use Biochem system that was developed, I think, in Eric's Go lab in Portland.
One question, I think this is still important, even though they seem to have answered the question themselves. The contrast of particles looks super nice for such a small protein. Just out of curiosity, could the increased signal to noise come from use of nanodiscs?
Yes, so we think so. So actually the contrast is better in our kind of hands for the protein that is reconstituted into nanodiscs. So yes, so we believe that in detergent, you have a lot of free micelles that also you have in your protein, so that may increase the contrast. But in our case, we didn't see much of the improvement in resolution in nanodiscs, so it's kind of that comparable.
All right. Next question would be regarding the open and closed states. So the open and closed states of the gain-of-function mutant, how did you conclude that there were no closed conformations in the gain-of-function data set?
I mean, it's a good question because when we did all the processing for the gain-of-function mutation, we didn't see any signs of the closed state. Of course, if you can try to and do like some additional processing and try to find something like 3D classification with like specific reference to squeeze this open closed conformation, maybe you can end up with something. But at least I think maybe we didn't see it, so it means that maybe the abundance, kind of the less than 10%, 5%, even less particles may end up in the closed state. And again, this is interesting because in single channel recordings, channels, they kind of flick between closed and open conformation, but it looks like that Cryo-EM conditions prime the channel to be in the open state.
So just a follow-up question. I was wondering if you showed the local resolution map of the structure, and in that it didn't seem like the top portion where the nanodisk is, there was not really a lot of change happening there. So would you still expect there to be a closed or an open state even after you do further processing?
So I just want to clarify the question. So you're kind of asking about can be open state in the set of particles that resemble the apostate, or what you're asking?
So you said that maybe further processing could resolve the open and the closed states in the gain-of-function data set. But I'm wondering, if you don't really see a lot of heterogeneity in that region, would further processing still help?
So this kind of a hypothetical question. So I don't know the answer to this. Yeah. So I don't know. It's always kind of with the development of Cryo-EM tools, you always kind of I expect that if you go back to your previous collections that were collected like 3 or 5 years ago, you can squeeze more and more and more from those data sets. So right. I don't know, but maybe with the development of the Cryo-EM tools, it's going to be easier to kind of classify those low abundant, low populated states.
Yeah. Next question is, did cholesterol remain bound when NTB were or NS were bound?
Yes, so we always see the cholesterol is bound there. So there is no competition. So cholesterol is close to the binding site, but it's kind of touches from the top. But it's a little bit kind of different. One thing that we observed, so when we used very specific detergent called digitonin, it also worked for our like extraction. It looks like the digitonin kind of substitutes cholesterol, but only in this case. In all other cases, we always see cholesterol bound to the protein at the same spot all the time.
One question is about the lower region of the TRPM7. What is the significance, the structural functional significance between the 2023 versus the 2018 structures?
Yes, so this is like perfect question, and we actually don't know I don't have like a good answer for that. What I know for sure is that for the naltriben activation, the binding actually occurs in this kind of middle part of the channel, but that are also kind of flexible. But for the lower part, I can speculate that we use the truncated version of the channel without kinase domain. I speculate that the lower part of the channel, if this kinase domain is present, it is involved in this kinase domain coordination. But I don't have any proofs.
All right.
My speculations.
Along the same lines, what is the role of cholesterol in TRPM7 channel? It's a more generic question. Yeah.
So there are some data about cholesterol, and it looks like that it kind of potentiates the channel. So it doesn't activate it, but it helps to activate it. But again, there are only like a couple of studies that show this, and we're still not sure what it does. We are thinking how we can test it. So you can deplete the cells from cholesterol and try to solve the structure and see what happens. Maybe it go like in different conformation. We can try to mutate residues around the cholesterol binding site if it is like indeed the binding site, although those there is no like specific binding, looks like just kind of hydrophobic, like leucine, valine, isoleucines. So the short answer, we don't know what it does, but we speculate it is functionally relevant, and it potentiates the channel function.
Sort of a follow-up question: does cholesterol bind to other TRP channels too?
Oh yes.
Okay.
Yes. So there are there are channels where cholesterol is absolutely required. Again, this is kind of in my experience. For example, TRPM6. So you can see not one, not two, but 3 different spots for cholesterol binding. And if you don't add cholesterol or cholesterol homologous when you do protein purification, you will end up with 0 yield. So yeah. It is very important for some channel, but for some TRP channels, it looks like it doesn't do much.
With respect to data processing, why shift between CryoSpark and Reliant when CryoSpark can do pretty much everything that Reliant can do?
I absolutely agree with this. That CryoSpark basically can do everything, and Reliant. So again, this work was done about like 2 years ago. I mean, we started this processing, and we decided to kind of stick to the same processing style. But again, in my experience, that Biochem polishing in Reliant works really well. And it works, in my experience, way better than local motion correction in CryoSpark. Again, in my experience. So although you sacrifice about several days to run this Biochem polishing, but you will end up in better resolution. So that's why I prefer to still use Reliant. And again, back in those days when we started those projects, the 3D classification in Reliant worked much better than in CryoSpark. Although CryoSpark kind of now it's they're close. I think they're even in 3D classification right now.
Does the N1098Q mutation affect the affinity or effective concentration for naltriben activation?
Yes. So yeah, it shifts the. So you need to add more high concentration of ligand to inhibit the channel. So yes, there's a shift.
There are 3 more questions. Did you have any problems getting enough protein for gain of function mutant?
The question is about the purification, right?
Yeah.
Yes. So we actually had like a lot of problems with this mutant. Why? Because again, as I mentioned, that TRPM7 is constitutively open channel. So it's not like always open, but it always like pretty regularly, like let's say like 1% of its time. Let's say I'm just making up this figure, this number, but let's say 1% of all the time it is open. And this is a big problem with protein expression, because as it's constitutively open, and when you express the protein for 2 or 3 days, the cells, they die because of the mineral overload, we believe. So they you need to have some tricks to inhibit the channel. And we actually added ruthenium red. This is the channel blocker. It is not specific, but it is a channel blocker, and it also blocks TRPM7 channel for protein purification. And we had to add like extra amount of this inhibitor when we did protein expression for gain of function mutations, twice as much at least, to make to give the cells a chance to survive while they were overexpressing the channel. So yes, we had a lot of problems. And on top of that, even with this trick, we end up in relatively low yield, and we were not able, there was not enough protein for us to try to do non-adhesive constitution. But luckily, detergent worked well for us.
Did you see big changes in the structure in GDM and digitonin since it substitutes cholesterol?
No, we don't see any changes if I'm talking about the apostate. So the only difference that we see, and also I mentioned this, so for some reason, when we added agonist naltriben to the protein in detergent, the protein didn't open. This is the only difference. But apart from this, absolutely same conformation, same lipids.
Do you have any hypotheses about the interaction of TRPM7 and the CNNM magnesium transporters?
So it's better to ask our colleague Vladimir Chubanov about this functional stuff. So yeah, I'm not an expert in this field, but he's like leading expert in this physiology of TRPM7 channels. So sorry.
Well, we can invite.
I'm not qualified to answer this question.
We can invite your colleague for a talk next time.
Oh yeah.
One last question. Have you tried to work with the TRPM7 where the kinase domain was kept?
Yes, we tried a lot. So the long story about this project, this was the first project that I was assigned when I just joined the lab. It was like 4 years ago. And the first idea was, okay, the structure was solved with truncated version already by that time. So what can we do? Why not try to do the full length with the kinase domain? And I spent a lot of time doing this. And the yield was so low, basically there was no yield. So it was not feasible to get any Cryo-EM data from that. It looks like that, and on top of that, it looks like that during protein expression, this kinase domain is cut off from the channel. So basically, whatever you do, you still will end up with the channel that lacks kinase domain. So there should be some additional tricks how to express the channel with kinase domain included.
I have a quick question, just from the observation when you were showing the motion of the channel in the those two kinds of motion, especially mode 1 there. It looks as if yes, it obviously the C4 breaks down, but perhaps C2. That is, that it still looks like both of those modes would allow a C2 symmetry Or is it maybe just the fact that you tried to enforce C4, and this is what you get? Have you C2 symmetry might do a little bit better than C1, but what's your take on the amount of symmetry that might be in here left over after C4 goes away?
Yeah, so this is excellent question, and I want to warn everyone who uses CryoSpark and run this variability analysis to kind of get inside whether you can see some specific movements in your protein. So in my experience, when we did this 3D variability analysis in CryoSpark, sometimes you got some fake movements. So although we can see it's kind of like flickering between like two like C2 conformations, like from like diamond shape, like lower part, I'm still not sure whether they exist. So I think this can be just artifact of variability analysis. And why I'm saying so? Because we for different proteins, we see some movements that were present in our protein, but those movements were fake. And we know for sure that they were fake. It's hard to explain why, but basically my warning is, so please don't overinterpret these movements in the CryoSpark. And answering the question, maybe like C2 symmetry will help to get better resolution. So what I think is happening here is that, so first of all, we have like two conformations. You can see that we also have this like coiled domain here forming laser pointer. So we have like a lot of different conformations in the lower part. First with coiled domain and without coiled domain. And what else is happening, at least this is my impression based on the like C1 symmetry refinements, is that sporadically the MHR domains, the lower part, like any of them can be just unfolded. It can unfold. So it means kind of, it can be like 2 unfolded, 1 unfolded, 3 unfolded. So, and we can see this during 3D classification. So I don't think this is like C2 symmetry mostly. I think it's just kind of movements and heterogeneity in each individual subunit that can be expressed in this like variability analysis in CryoSpark. I hope this answers the question, at least part of it.
I fully agree, and that there are now other ways of looking at these motions, including the Gaussian mixture model that comes out of EMAN. It's a different way of modeling the motion. So maybe those things would work too.
Yep.
Did you happen to put a mask on the regions where the variability is the highest? That's what I've usually done. I've just put a mask. Initially, I do a 3D variability on the whole structure, but then I localize it by just putting a mask on that region, and then separating them into clusters. And those clusters, if they end up giving me a high enough resolution structure, then I'm more inclined to believe that this could be true than not just.
Oh yeah, I mean, I absolutely agree. So at least what we observed for these like lower parts, these MHR regions, is that those are only like rigid body movements. So the kind of, what I mean, there is no kind of there is no flexibility in those regions. It's just like the whole region of the like each subunit moves and rotates. So we did this analysis, at least kind of part of it.
It seems like we just gave you a bunch of homework, but that was a really super useful discussion, I think. So thanks so much for sticking around and getting to those last questions. That was fantastic. I'm gonna go ahead and stop our recording.